Professor Redegeld is an associate professor at the Division of Pharmacology at Utrecht Institute of Pharmacological Sciences, Faculty of Science, Utrecht University in Netherlands. He is editor-in-chief of European Journal of Pharmacology. His current research interest involves the molecular mechanisms of cellular activation in chronic inflammatory diseases, with a strong focus on mast cells. He is a member of multiple national and international associations. He has a very special interest in mast cell. And today he will be talking on the Eosinophil Enigma. Over to you, Prof.
Okay. So is my presentation visible now?
Yes, it is.
Yes, yes.
Yeah, okay, perfect. So I'm really happy and glad that we, especially Kunal, could organize the meeting as such so far. And I'm really happy to contribute to the meeting with this presentation. I'm going to talk about EGIDS. I will tell you a little bit about eosinophils themselves, where they come from, how they mature, where you can find them in which tissues, and what function they may have. Then I go on to some of the EGIDS, and especially I end up talking about EOE, which is a disease we are actively researching at this presentation. Thank you very much for the invitation. And now I'll start the next slide. Hopefully that will work. Yeah. You are here, and I'm here in this tiny little country in Europe. And this is a very small country. We have only 17 million people in the Netherlands living, and although it's still quite crowded, especially in the area of Amsterdam, Rotterdam, and Utrecht. Let me see. I do not see my slides moving. Yeah, there we go. I'm in Utrecht, and this is in the middle of the country. It's quite congested with all kinds of public transportation, especially nowadays. And Utrecht is a very beautiful city. And of course, you are probably familiar with all the pictures of the Netherlands with the tulips, which will be visible in spring. We have very nice cities, Amsterdam, Rotterdam here, and some castles as well. And currently we are experiencing a lot of snow. This is in my front yard, so my neighborhood, and it was snowing like crazy. And we got about 30 centimeters snow last week, and yeah, it causes a lot of troubles with transportation at Schiphol airport and etc. etc. So, but it was also a very pretty sight for us, and now it's melting, so the sight is less beautiful at the moment. I'm working on the Utrecht University, which has been established in 1636, and it consists of the university itself, but also the Utrecht Medical Center. It's a big academics medical center, and the Princess Maxima Center, which is specialized in treating children with cancer. We have about 150 companies on campus, startups, over 70 in life sciences, and we have daily about 30,000 people coming in working in the university, and we have about 55,000 students. So it's quite a nice place to work with beautiful buildings. This is a student building, for instance, Casa Confetti. And this is the building I'm in. And actually, this corner room is mine, which is, I have a very pretty sight over the campus. And here in this wing, we have the laboratories, all the laboratories. I missed one slide, but okay. I want to continue now with the scientific presentation. And I want to talk about, and I will change my pointer if that's possible. About eosinophils to start with. And what are eosinophils? These are these pretty cells with a lobular nucleus. That's how you can identify them. And you usually have a pinkish color when you stain them with hematoxylin and eosinophil. They are granulocytes, and they may circulate in the body, in the circulation, but mostly are present in tissues. They are involved, as you probably know, in parasitic defenses, allergic inflammation, and tissue remodeling. Normally, you don't find so many eosinophils in the GI tract, but in case of inflammation, they could travel there and help to do some damage in the organ. Because they contain cytotoxic granules, and that really can damage the tissue when they are activated excessively. The eosinophils are mainly involved in type 2 allergic immune responses, and the key cytokines in this story are IL-5, which is an eosinophil growth factor and survival factor, but can also stimulate eosinophils, and IL-4, IL-13, which are involved in tissue inflammation, creating tissue inflammation, and thereby attracting the eosinophils into the tissue. One of the main chemokines to recruit eosinophils to the GI tract and to other tissues are eutexins, and of course IL-5 will also help in this. The eosinophils develop mainly in the bone marrow, but not exclusively. They can also be generated in situ. And this is a picture of the airways, because it was a very nice illustration, and I will enlarge this. And normally bone marrow go from stem cells to myeloid stem cells, and then into eosinophils, and then mature to fully developed eosinophils, and then they are released from the bone marrow and can be attracted to tissues, like here. But under certain circumstances, also the eosinophils and the stem cells can be attracted to the tissue and there develop into mature eosinophils. And that's done by the release, if you have damaged epithelial cells, they release alarmins. They can activate the ILC2 cells, which are producing IL-4, IL-13, IL-5, and these are interacting with basophils and mast cells, which can also induce the production of IL-4 and IL-13. And these cytokines can help to drive the progenitor cells into fully matured eosinophils. So you don't have to get the full mature eosinophils right here in the bone marrow, but also locally they can be produced. Now, how do if we go from the bone marrow to the tissue, usually when they enter the circulation in bloodstream, the increased expression of adhesion molecules like M1, VCAM, and METCAM will tell the circulating eosinophils, oh, here I should stop. They don't roll anymore. They stop rolling, and then they are egradicating the bloodstream and go into the tissue. And the drivers here are IL-5 and eotaxin. And they can go into all kinds of tissue. We can tell that the main driver, regulator of eosinophil development is IL-5. If you block the binding of IL-5 to the IL-5 receptor on the eosinophils, like with Luzimab, you can catch here the IL-5. You don't have much of a development of eosinophils anymore. However, there is literature around in mice models with an IL-5 knockout that there are still eosinophils in tissue. So there is other factors also involved in maintaining kind of a basic eosinophil number in different tissues. Currently, in the latest literature, people are talking about different subtypes of eosinophils. There's not only one subtype, but there is resident eosinophils and inducible eosinophils. And these may be the inflammatory eosinophils, and these may have a role in the homeostasis of tissues. And for human, these are the cell surface markers which are present on the resident and the inducible EOs. So you can distinguish them based on the phenotype, the cell surface receptor expression. Same is true in mice. They are a little bit less complicated. So what do the eosinophils in different organs? If we go to the thymus, the half-life of eosinophils is around 6 days, and they may help with negative T cell selection and clearance of apoptotic cells. Not so much damage is caused, of course, because the thymus is a very important organ. Also, mammary glands, they have a role in the homeostasis of the tissue. I have some, yeah, okay. In the gastroesophageal tissue, they may also have a role in the homeostasis, maintenance of plasma cells, IgA secretion, development of patches, etc. But they can also have a role in the pathogenesis, like I will talk about later on the eosinophilic esophagitis and gastroenteritis. And we can go on with bone marrow and blood. They can cause hypereosinophilic syndrome, and they may have a role in atherosclerosis and thrombosis. Adipose tissue, perhaps a role in inducing diabetes, but they also have a role in the creating sensitivity to insulin and glucose tolerance. Lungs, same story. They can induce lung inflammation or help. Yeah, they are involved in lung inflammation and eosinophilic chronic rhinosinusitis, allergic rhinitis, etc. So they have kind of a double role. And I will mainly talk about the role in inflammatory reactions, of course, because that's probably most important to you. In the granules, you find, or in the eosinophils, you find granules, and they have a two compartments, a kind of a crystalloid part where myelin basic protein is the main component, and a more soluble part where cytokines and enzymes are contained. Eosinophils also release vesicles, and they can contain small secretory content, and they have chemokines and possibly some cytokines inside, and they are characterized by these vesicle associated membrane proteins. These are snares-like, so they are actually involved in binding to other cells. It is shown that eosinophils can produce and release quite a bunch of interleukins, chemokines, growth factors, and others. I must tell that the amounts of these interleukins are not comparable to what, for instance, T cells can produce, or ILC2 cells. But in their direct environment, of course, they can have a huge effect on other cells. So what is the communication of eosinophils with other cells? What may they do? Eosinophils, by the release of eosinophil-derived neurotoxin and peroxidase, they can help to recruit and activate dendritic cells. This chemokine CCL6 can impair the homeostasis. Here the neutrophil can be activated by different cytokines and chemokines released. It can recruit Th2 cells. It can help to polarize by the release of different cytokines the T cells locally. It may suppress Th17 differentiation and proliferation, and help the survival of CD4 T cells. Now I can go on. So it has a lot of effects of neighboring cells, which are usually also present in the inflammatory soup we have in the in the tissue of an affected organ. Again, some of the regulatory roles, it inhibits the Th17 expression. It can have an effect on the regulatory T cell induction, and it has regulatory cytokines and other factors like galectin-10 and TGF-beta. So it's a kind of a versatile cell in the immune system. But I must also say the clear role of eosinophils in the pathogenesis of different diseases is not always very outlined. We still don't understand their role. What do they do? Are they necessary in the pathogenesis? And I'll come back to that later. If we look at the spectrum of eosinophilic diseases, we can think about eosinophilic asthma, the EGIDS, of course. This is a granuloma, EGPA, chronic rhinosinusitis with nasal polyps, where there's a lot of eosinophils infiltrating, and of course, hypereosinophilic syndrome. I will talk about these diseases further on. And EGIDS are chronic immune-mediated diseases characterized by an eosinophilic inflammation in the GI tract. And it can go from the esophagus to the colon. Immune mechanisms in these EGIDS, they resemble an allergic response. Usually there is a breach in the epithelial barrier, and thereby allergens and pathogens can enter the tissue, and then they are recognized by DC, local DCs, which are activated, and then on its turn, they activate the T cells. Under the wrong conditions, when IL-4 is present or IL-13 is present, these Th0 T cells develop in Th2 cells, and they stimulate B cells to switch their immunoglobulin repertoire in the production of IgE instead of IgGs. IgE can bind to the mast cell, and upon contact with the allergens again, you get cross-linking of the IgE receptor and activation of mast cells. And you get histamine, serotonin, leukotriene, cytokine production and release, which have effects on the smooth muscle fibers, but also on many other cells. With the help of this IL-5, also eosinophils can be activated, but they can also be triggered by these alarmins which are released. IL-13 on itself can also damage again the epithelial barrier. So you get a vicious circle where cells are being activated by the production of alarmins, and then again you get more damage of the epithelial barrier, again, and more stimulation by different allergens, pathogens, etc., etc. So this is a classic allergic response, whereby the main actors are probably IL-4, IL-13, and these alarmins, IL-33, TSLP, which initiate at least the responses locally. When we look at eosinophilic colitis, what do we see? It's distinguished by clinical manifestations, including abdominal pain, diarrhea, weight loss, loss of appetite, bloody diarrhea, constipation, mucosal lining damage, which is caused by the immune response, and finally also impaired nutrient absorption. EOC involves abnormal hypersensitivity, where infants may have an association with allergens, and maybe adults have a more DTH-like response where T cells are involved. DTH is a delayed type hypersensitivity, type 4 allergy. It is characterized by the infiltration of eosinophils into the colon, accompanied by the increased levels of IgE and IL-5 in the bloodstream. So you could actually have a diagnostic tool to be used for the measurement of at least IgE in the blood. Eosinophils, an increase in eosinophils that you do not much see in these patients. So you have to go locally, and endoscopic examination reveal erythema, loss of vascularity, and lymphonodular hyperplasia. And usually you're also taking biopsies to look at the presence of EOs in the tissue. And you should have at least hundreds eosinophils per high power field in the cecum and the colon, at least 84 in the transverse and descending colon, at least 64 in the sigma and rectum. Whereas the normal counts in the large bowel is 8 to 30. So, but it's a patchy disease, so you should take more than one biopsies to count these cells, because you may just pull the wrong side from the organ. Now you do see crypts. I'll show you pictures in the next slide. Alteration in the crypt structure, formation of eosinophils in the epithelial layer, presence of eosinophils in the muscularis, etc. So there's infiltration of eosinophils all over. It's crucial to distinguish EOC from other conditions like an infection, perhaps a parasite is there, HES, ulcerative colitis and Crohn's disease, and connective tissue diseases like Sjögren's syndrome, and other like systemic mastocytosis. You may also see in the bowel, in the colon, some EOS appearing. This is a picture of what you may see when you do a colonoscopy. You see patchy erythema. You see strictures like this, and you see a lot of eosinophils appearing in the tissue. For eosinophilic gastritis, it goes along with vomiting, abdominal pain, diarrhea, malabsorption, protein-losing entropy, anemia, and failure to thrive because probably the malabsorption, and melena. The EOG patients often exhibit increased blood counts of eosinophils. So there, an increase in the eosinophils might be predictive for the disease. But again, you have to look locally whether it's really the case here. So the diagnostic criteria for EOG include the presence of at least 30 EOE, eosinophils in a high power field, in at least 5 biopsies, or 5 different sites in a biopsy, or at least 70 eosinophils in 3 high power field biopsies, high power field specimen from biopsies. That goes along with different numbers for the duodenum and ileum. Usually, patients with EOG have allergies, and they have IL-5 positive Th2 cells. And when you trigger them with food, you have an increased development of the Th2 cells in the tissue. And if you take a biopsy and incubate that with food, you may see an increased production of IL-5. EOG has a distinct transcriptome with minimal overlap with EOE, which is a little bit more up the gastrointestinal tract. Here you see a colonoscopy of a gastroenteritis patient before the treatment of with corticosteroid prednisolone, and here as well. So the inflammation, you see here the dark things, they disappear. You get a cleaner tissue vision. That's also true. I believe that's here. With the histological staining with H&E before and after, and here as well. So to compare these diseases, and I will end up later with eosinophilic esophagitis. The prevalence of the disease for EOE is around here up to 50 patients per 100,000 individuals. Eosinophilic gastritis, enteritis, 6 per 100,000. So the numbers are lower, and eosinophilic colitis even lower here. And it means that there is not that many patients around to investigate the clinical symptoms and the drivers of the disease. Eosinophilic esophagitis is nowadays quite in focus, because we've seen definitely that during the last 20 years there's a huge increase in numbers of affected subjects. So there's the numbers are higher and they're easier to approach. For all, there is diagnostic criteria, but just counting the eosinophils in biopsies taken from the organs, as I just explained to you. The clinical features we have gone through, endoscopic features. I will show you some endoscopic features of EOE in a minute. And here we have seen the pictures already. Okay. How to treat non-eosinophilic esophagitis, eosinophilic gastrointestinal diseases, the non-EOE EGIDS. You could use corticosteroids, prednisone, for instance, topical corticosteroids, or at least in a formulation that stays opens earlier than at the distal locations. Acid suppressors, proton pump inhibitors, PPIs, histamine receptor antagonists, Crobolin, mast cell stabilizer, ketotifen. They don't work so well in most of the patients. Then we can down the leukotriene inhibitors may work. The small molecules, anti-inflammatory molecules, and of course biological mediate antibodies against specific targets. I will come to that also a little bit later. And then new drugs are on the horizon, like SIP receptor modulators, Janus kinase inhibitors, antibodies against other cytokines not mentioned here, antibodies against KITs. I'll come to that also in a minute, which deplete mast cells, and calcineurin inhibitors, which is part of activation cascades for different receptors. Here a picture, like IL-5 inhibition is much in focus, of course, either by trapping soluble IL-5 or blocking the receptor. Imatinib is a BCR inhibitor, protein tyrosine kinase, which is involved in the signal transduction of different cytokine receptors. And then we have omalizumab, which prevents binding of IgE to the low affinity receptor, because the eosinophils are not expressing any high affinity IgE receptors, and some other molecules may be blocked, like the JAK1, JAK2 kinases. I want to move now to work we have been doing on eosinophilic esophagitis, a very interesting disease because it's a little bit different from the other EGIDS. There is definitely, they have in common that there is eosinophils infiltrating in the tissue, but other mediators are probably a little bit different. So touch upon that in a minute. For eosinophilic esophagitis, they say that dysbiosis of the microbiome might be involved. There is some genetic differences visible in different families developing eosinophilic esophagitis. So there's a gene component here. It all leads to inflammation and tissue remodeling. And what the trigger is not always clear. There's definitely impaired barrier function, whereby the local, for instance, food components can enter the tissue of the esophagus. Reflux may play a role because that's damaging the barrier as well. And we know that for most of the patients, allergens are part of the cause of the disease. As said, there is an increasing prevalence of EOE. we see EOE is mostly developed in males around when they are around 20 years old, but also children with a lower age may develop EOE. But that's a little harder to diagnose, of course. Usually they don't thrive, and then they go to the doctor, and then some tests can be done. But usually it's kind of taking away some food components because there is not so much biomarkers available. I'll show that in a minute. It's not only a disease in the Western world, it's also developing and increasing in Asia, because now numbers become available, and you see also an increased prevalence of EOE in Asian patients. Endoscopic signs. This is a healthy esophagus. In EOE patients, you see edema. You may see furrows, like here, rings, strictures, exudates, and here at the end you can also develop crab paper esophagus. This is very sensitive tissue in the esophagus. Again, pathogenic hallmarks of EOE are barrier dysfunction. There's type 2 inflammation, and when the inflammation goes on and on and on, you get remodeling of the tissue, creating more problems to the patients as well, because the swallowing is more difficult and food gets stuck easier in the esophagus. Same thing, eosinophils are present. You see basophils, you see mast cells, which release all kinds of mediators acting upon smooth muscles, fibroblasts, neurons as well. And usually there is a decrease in barrier proteins, which keeping the epithelial cells tied to each other, and increases in, for instance, proteases or antiproteases, and decreases in antiproteases. So all in all, it's an epithelial barrier disease, I can say, leading to local inflammation, local allergic inflammation. And the diagnosis of EOE is difficult. You have to do an upper endoscopy and then look at the lining of the esophagus, see if there's any difference, swelling, horizontal rings, vertical furrows, narrowing, white spots. It's usually accompanied by taking biopsies from the esophagus at different places, and these will be examined for the presence of eosinophils. Instead of doing biopsies, you could also swallow a sponge and then leave it a little in the stomach, and then pull it out again, and then it takes a long tissue which can be examined for the presence of eosinophils. Blood tests, they don't work. They are not predictive. What we do usually with food allergy or other, and maybe even with the other EGIDS, look if there is an increased response to food allergens. That's not predictive at all. So measuring IgE levels and eosinophil counts doesn't help. We've worked a little on the diagnosis of EOE because it's really difficult for patients to find out what is causing the response in the esophagus. Skin prick tests, atopy tests, they also don't work. So they are really they are really willing to find what is causing the disease, because it's also a very psychologically affecting disease. And suddenly you can have this your esophagus swelling, and then you food got stuck, and maybe you have to go to the hospital to get it out. So what we have done is to see if we can use esophageal tissue from a biopsy, and then add the different food allergens. Usually we took some of the common food allergens like milk, egg, etc. These are quite common allergens for and wheat in the Netherlands, and then challenge this tissue ex vivo in a reagent tube, and to see what happens. So we did an ex vivo food challenge of the esophageal biopsies and see what kind of response we got. We had in this study 12 adult EOE patients and 6 non-EOE patients. They came for other reasons to the hospital, but biopsies were taken to see what was going on, and we stimulated them with 6 foods or saline. So 3 general foods: apples, cows, milk, and wheat, and some foods based on the patient's clinical history. Some people say, okay, I'm allergic to beer, a particular kind of beer, so we put that in, etc. And some are for tomato, and others for milk. And then we, after incubation, these different biopsies, we analyzed the inflammatory profile using an array, cytokine array, which was run on a flow cytometer. So this is the protocol. We had the biopsies, incubated them for 24 hours with a single food. So we had more biopsies, parts of the biopsy for a single food, and then measured the cytokines and chemokines. This is what we got in non-EOE patients and in EOE patients. We had 3 different cytokines which are really involved in allergic responses, and they were significant: IL-5, IL-13, and MCP1 and they were significantly increased in EOE patients compared to the non-EOE subjects. Total IgE was also increased in these patients. IL-6, IL-8, more general cytokines were increased, and then IL-10 and TNF-alpha didn't reach any significance. So what to do with these data? We had a statisticus in our group, and he put these data we had in a machine learning routine, and also included the SPT and serum IgE results we got from the patients. Yeah, then with a lot of mathematics, he analyzed the data and said, okay, these IL-5, IL-8, MCP1, and TNF-alpha and SPT2 may have a better prediction than just measuring the SP2 and the serum in the samples in the patients. So this is what you get for response curve for SPT. Same for IgE. Yeah, it's 50-50. You can measure it, and then you can flip a coin. That's possible. So it doesn't help you to generate any prediction diagnostic value. In our case, with the 4 different cytokines, IL-5, IL-8, MCP1, and TNF-alpha, we already got an increased prediction. It went up to 0.64. It's, of course, not good enough, but this is maybe a way, we are exploring that now, if you measure more produced cytokines, chemokines, and other factors to delineate whether a food factor is involved in triggering the disease in the tissue. And these studies are ongoing. So we can summarize this part that EOE esophageal biopsies cultured in ex vivo with fluid components actively produced type 2 and pro-inflammatory cytokines, chemokines, and total IgE. I didn't show you, but IL-9 levels, which are important in triggering also mast cells, were markedly increased in these biopsies in 7 out of 10 patients. It's interesting. And the 4 cytokines I showed you in the previous slide were more predictive than SPT and serum IgE measurements. And this is, of course, also a not that invasive way because biopsies are taken always for the examination of the tissue. If you take a few biopsies, 2, 3 more, you can do the analysis of the cytokine production. The pathophysiology of the disease. Yeah, there are key factors where you may take action on to reduce the disease and perhaps completely resolve the disease. And the suspects are, of course, cytokines like IL-4, IL-13, IL-5, acting on eosinophils. You may also look into TSLP and IL-33. You can look at mast cells because the disease is accompanied by mast cell activation, although systemic IgE is not predictive, but you do see a massive mast cell infiltration in the tissue and also activation of these cells. So you could go after these factors. And perhaps a question to you, what would you target? Is there anybody in the audience who could tell me what's the best way to target this disease? Should we take out the eosinophils?
No, either the alarmins or the 413 cytokines. So
Yeah, the alarmins are very interesting. As far as I know, there is studies around, especially with anti-TSLP in EOE, but it's not conclusive yet. So they are in phase 1, phase 2 stages. Of course, we have a cytokine which is blocking IL-4 and IL-13. And a biologic which is blocking those. We have IL-5 has been tested. There is drugs targeting eosinophils and mast cells at the same time. The anti-Cyclac-6 antibody. But I will show you some of them. So non-biologics are, of course, the most cheap way to treat inflammatory diseases. And you see proton pump inhibitors are tested, but they are only effective in a minority of EOE patients. And most of them are PPI non-responders. That's a pity. Local steroid treatments works quite well. It reduces symptoms. Endoscopically, the tissue regenerates. It's, of course, a cheap way. But there are also patients non-responding. And yeah, we don't know what happens if you treat patients for a very long time with local steroids. Because food is triggering the disease, you could also use amino-based nutrients, or even where proteins are fully digested in peptides, or even worse, in amino acids. This is not a food you want to take the rest of your life, of course. This is not. It helps to reduce the symptoms on the short term, but you cannot base your food on these amino-based nutrients. So they are rarely used. Elimination diets, that's also being used to distinguish the foods which are causing the symptoms. So you can start with a 7 food, 5 food, 2 food, or 1 food elimination diet, where you take out 7, 5, etc. components from your food which are suspected in inducing, and then you slowly reintroduce the food component again and see if the disease gets worse again. Then you know, okay, this might be a trigger. We should not use that in the diet anymore. But it's a long way. It takes at least a year, and sometimes it goes wrong that people accidentally take the wrong diets and eat something again, which was already eliminated as being causative. So, and then you're back to square one again and start the elimination diets again. But this is quite a good way, non-invasive way, at least. I don't know. For me, it would be invasive to take out all the nice food components, and probably for you as well. In that way, it's invasive in your life, but it's not invasive in the body. And then, of course, empirical elimination, that's based on what the patient thinks it is. And then if you take that away, then probably you don't have this this food, but you don't have any scientific evidence that it is causing the disease, of course. In the Netherlands, yeah, most of the problems are caused by milk allergy, and wheat. Yeah, the others are also playing, but it's difficult to tell patients that you have to eliminate these components and that the disease will be solved. That's, of course, not always the way. Biologicals for EOE are now being developed, and some are doing quite well. So we have the eosinophils, IgE, alarmins, and type 2 mediators. Let me see. We have here, so I will show you these 4. So anti-IL-5, anti-c-KIT, which is a growth factor, essential growth factor for mast cells. If you block this receptor, c-KIT, then you eliminate the mast cells from the tissue, virtually all. Anti-TSLP, and then the cytokines. So you would think EOE, eosinophilic esophagitis, eosinophils are having a great role. If you treat patients with mapeluzumab or benralizumab, lintinimab, they all deplete eosinophils from the tissue. This by binding IL-5, this by blocking the IL-5 receptor, anti-cyclic 8, by causing antibody-dependent toxicity to eosinophils. It binds the cyclic 8 receptor, which is an inhibitory receptor. All eosinophils disappear, and it doesn't do anything to the patient. Symptoms stay, and yeah, it's unbelievable. So eosinophils, some people say, okay, we should rename eosinophilic esophagitis, take out the eosinophil, because it's not doing probably any harm in the tissue. But you can use it definitely as a biomarker. You can, for diagnostic purposes. IgE, I would set my bets on IgE. Oh no, not on IgE, but on mast cells. IgE using omalizumab results in lower IgE levels in the patients and lower mast cell counts because it binds free IgE and it doesn't bind anymore to the mast cell, and therefore mast cells cannot be activated locally by cross-linking of IgE. In this case, omalizumab was tested in the EOE patients. It didn't lead to any clinical remission in the EOE patients. So we cannot use IgE. Although we also saw that when you incubate biopsies with food, you see increased production of local IgE, total IgE. It doesn't help. What's going on? Perhaps omalizumab is not entering the tissues so much, that there's local IgE production, and this might trigger the mast cells. I don't know. It hasn't been tested further. Anti-c-KIT barzolvolimab, just tested by Celldex. It leads to lower mast cell counts because it binds to the c-KIT receptor. There is no growth factor for mast cells anymore. You don't have mast cell survival anymore. You see decreases in the mast cell numbers, but unfortunately, no clinical remission in the EOE clinical trials they've published recently, I think in November last year. So also mast cells do not play a role. I still have some questions about that, but it's not a place to discuss that here. But maybe there is a role for mast cells because we have seen in our in vitro studies, ex vivo studies, that mast cells are definitely activated. And responses to we've done studies, and maybe I will talk about it during the conference where we injected food allergens locally in the esophagus, and we see an immediate response of the tissue. You see swelling, edema formation. So it must be triggered by a cell responding in an antigen-specific way, because not all food allergens we injected were causing this in the tissue. Anti-TSLP, tezepelumab, that's now being tested in phase 3 trial. So we don't know if that will work. I'm anxious to see the results because I think it might be a very nice alarmin to block, which may be crucial in the cascade, the allergic cascade. Then the only one which works, anti-IL-13, is now in a clinical trial yet. We don't know the results, but the only one which works is dupilumab. And it works very well in EOE patients. It blocks the IL-4 receptor alpha chain, and thereby blocks IL-4 and IL-13 signaling. It has been approved in 2022 for the treatment of EOE patients, and now it can also be used for younger, first adults, and now it's also approved for younger EOE patients. It's a monoclonal antibody which triggers one component of both the IL-4 and IL-13 receptor. And it's injected subcutaneously in a weekly fashion. So it blocks this receptor, the IL-4 alpha receptor, which cannot dimerize anymore with the gamma chain in case of the IL-4 receptor, and it cannot dimerize with the IL-13 receptor, whereby both cytokines are not active anymore. And these are quite upstream, the allergic response, of course. Here,
20 minutes late off schedule, so you'll have to finish it early.
Sorry?
We are running late, Frank. We're 20 minutes beyond the time we were supposed.
Okay, yeah. Now this is almost my final slide, so I will okay. I'll skip this. It's being approved and being used for difficult to treat EOE. We don't know about the long-term treatments, and of course, high costs are involved in this treatment. Unanswered questions so far. about eosinophils. Do we have really pathogenic subsets? Do we take them out with the different treatments? Is there reversibility of fibrosis, and what are the long-term depletion risks? Yeah, there are some research opportunities, of course, in Asian countries. Are the symptoms and the status of EGIDS similar to Western world? There is not so much information on that. Is there a different environmental, dietary, genetic risks involved? Clinical presentation might be different. So these should be observed and maybe also published in a proper way, so that we can learn if diseases are different in different parts of the world, and if we also have to take action to use different drugs to treat the patients. Key takeaways: eosinophils are central drivers of EGIDS. They mediate inflammation, tissue injury, and symptoms, and the diagnosis relies on tissue eosinophilia. This is the only reliable diagnostic way. And targeting eosinophils improves disease control, but it doesn't dissolve symptoms, as we have shown. I want to show the team I'm working with, especially Mirel Kleuskens and Marie Beck have done a lot. These are the technicians in my group. And we have a strong collaboration with Arjen Bredenoord. He's the gastroenterologist who did all the biopsies, etc. And their PhD students, Laura and Mandy. Sorry, I'm late. Thank you very much for listening, and happy to answer questions if they are still there.
So there are a few questions. Dhaiwat bhai is here.
Yes, please. Yeah.
Yeah.
So Dr. Abhishek Patil has raised a question that how often are this EGID associated with food allergy? Are they more commonly idiopathic?
No. Food is definitely a factor in these diseases as well. And you see, but with EOE, that they have comorbidities, and they usually people are food allergic, or they show other allergies accompanied with EOE. So there is also some evidence that immunotherapy may lead to EOE. So if you treat orally with allergens, it could also cause symptoms in the esophagus. But in most patients, you do see that food is a trigger. Yeah.
Thank you so much. Someone has asked a question. When to suspect EGID? You have already told that, but could you please briefly tell us about it, summarizing it maybe?
Sorry, when do you expect?
When to suspect EGID?
Yeah.
When do you
In which kind of clinical scenarios, I mean?
Yeah. So I'm not a physician, but usually you see different symptoms, as I pointed out in one of the slides, depending on where the disease is occurring in the gastrointestinal area, bowel, in the colon. You see that people are having problems, pain, etc. They don't thrive very well because there's maybe very bad absorption of food components because of the damage and the restructuring of the tissue in the intestinal tract. So there are different symptoms to look at, depending, and the symptoms differ a little from where the disease is happening. So look, with the EOE patients, they have pain in their breast. They feel that something is stuck in their esophagus. Swallowing is more difficult. So these are symptoms you have to look at.
Thank you so much.
I don't have yeah, sure. Yeah.
Yeah.
Is there any further questions? I don't know.
Yeah, I think I had a question that so there's this debate that goes along that how much do you eliminate? Do you eliminate 2 food diet, just milk and wheat, or you eliminate the 4 food diet or the 6 food diet in the
Yeah.
empiric elimination? And how long do you eliminate? Do you
Yeah, I think there are studies which compared the 1 or 2 food elimination with 5, I think, and they were both as predictive as well. So the less you eliminate, the better it is for the patient, of course. It's easier to adhere to the diet. You don't make easy mistakes, I think. Yeah, so and for I said in the Netherlands, milk is usually a big factor. I don't know that will of course differ in different countries. It could be peanuts in other countries or whatever. What is in the regular diet?
Yeah, in India, it's again milk and wheat, but our diet is so much based on milk and wheat that elimination diets often don't succeed the way we would want to, because patients
Yeah.
probably would want milk or wheat in their diet.
Yeah. That's the problem. You take away some essentials from their daily life, and it's really hard then to follow up your instructions. Although it would be good for the patient. Yeah.
Yeah, I think we've gone half an hour overboard. Thank you so much, Frank, for coming and for taking this wonderful talk. And
Sure.
I think I forgot to introduce our chairs and co-chairs today. So Dr. Himanshu Pathak is a senior rheumatologist and immunologist from the city of Baroda. And Dr. Dhaiwat Shukla is again a rheumatologist. He was a senior of mine when I was training in my medicine days and
Uh-huh.
practices in the city of Ahmedabad. And we are very excited to welcome you next month. It's just a month
Yeah,
to
it's yeah.
conference
I'm really looking forward to that, to attend the meeting and participate in it. Yeah.
so much for the elucid and detailed talk on eosinophils and their role in gastrointestinal disease. The only
Okay.
sad thing about this is that we don't have dupilumab in India currently. We hope we'll get.
No, yeah. Yeah, and it is extremely expensive way, and we don't know if you can taper off the pilibab. Perhaps you have to use it lifelong, and I think in the Netherlands it's about 20,000 euros per patient per year, and that's insane. If you have to take that your lifelong, you start at your 20th year. That's yeah, that's unbearable. But that's also a discussion which is going on in the Netherlands. All the biologics are very expensive, and some of them are just increasing the lifespan a little. For these patients, you could say, okay, it's not a deadly disease, so they don't die of it. But the quality of life will increase tremendously. So maybe we should put them on such an expensive drug.
Yeah.
But I may expect that if you have a normal esophagus, for instance, after treating for 1 year or 2 years with dupilumab, you may switch to another drug which is less expensive, and perhaps that will work. But these protocols haven't been investigated yet. So.
Yeah. Thank you so much again, Frank. And I think
Sure.
we'll call it a night today. So
Okay.
take
Wishing
care.
you a
Yeah.
very good weekend all, and hope to see you in February.
Same to you. Enjoy the snow.
Okay. Bye-bye.